This is the INAD PLAN Opportunities for Families in Research session. We've got two fantastic speakers. And what we're going to do is first have Allison Gregory speak, and then we'll have some time for questions. And then Dr. Paul will speak, and we'll have time for questions for him. Feel free to write any questions you can think of along the way in the chat box. I'll be monitoring, and so will Allison and Dr. Paul. And no question is too stupid. This is the time to ask, because you've got the experts on this panel. So as I said, our first speaker is going to be Allison Gregory. She has been a board certified genetic counselor since 1996. She completed her undergraduate work at Stanford University in human biology, and then went on to receive a master's in human genetics from the Medical College of Virginia. So she's now working as an assistant professor in the Oregon Health and Science University at the Department of Molecular and Medical Genetics. She's been working for 18 years in this space, studying neurodegeneration with brain iron accumulation. So she develops, implements, manages clinical research projects, assists with writing scientific papers and grants. And I've already seen her speak. She's very open and friendly to questions, and I look forward to her presentation. So without further ado, we have a recorded presentation, but again, Allison will be here for questions.
Good morning, I'm Allison Gregory from Oregon Health and Science University, and I've been given about 10 minutes to tell you about our PLAN-ready natural history study, including both what it is and why it's important. So I'm hopeful that most of you already attended or joined in with the natural history talk earlier this week. So I'm not going to launch into a bunch of definitions other than to say that natural history studies are important because they lay the foundation for clinical trials in our mind. And what I mean by that is, in a clinical trial, you usually have a small set of outcome measures that you measure on each person to see if your intervention is making a difference. And picking that small group of outcome measures is very important. If you pick the wrong ones, that can be problematic. It could lead to a trial failing when really the intervention was effective. So we want to have very solid natural history data to support any future clinical trials in PLAN. Our study itself includes subjects or people with both the infantile and juvenile forms of PLAN. At this point, we don't have the adult dystonia Parkinsonism group included because they're quite different, and trying to be consistent with sets of questions and things would be hard. There may be opportunity for that in the future. So at this point, there's sort of two branches to the study. The one that's highlighted here is the one that's more relevant for families and guardians. And then the other participant is a healthcare provider, which I'll explain in a little bit of detail. As far as parents and guardians go, everything is done online. And we hope that makes it easier for you. It means that you have a window of time every 3 to 6 months to answer a set of questions online. And you can do that at midnight when everybody's asleep, if that's best. You can do it in the morning, you can do it in parts. We're just hopeful that you'll be able to get it done. We ask about developmental milestones. So you're familiar with those things related to motor skills, social development, speech and language. And then we also ask about what we call disease milestones that are specific to PLAN, and I'll explain those in a minute. And then for each group, we also have a patient-reported outcome measure, which is a standardized published measure. I'm sure you've taken these in the past. Something like an intelligence test would be one of those, or a Vineland scale, but they're standardized and they're normed, so we can use them to compare between groups and within groups more reliably. There is also a role for a healthcare provider in answering some more detailed questions for us. And then we also try to get medical records from an earlier point in time for each participant. So we have sort of an entry point, maybe before the onset of their first symptoms. So a little more detail about what we ask families to do. It's serial data collection, so it's done in real time, and every 3 months for kids 5 and under, or every 6 months for children 6+, we ask you to do an update. We also have designed a way for families of deceased children to participate, and some people have felt really strongly that they wanted to do that. So if that's of interest to you, you can let me know, but that does involve just a single entry online. We collect it in real time to capture both the attainment and the loss of developmental skills that I described previously, and then also what we call disease milestones. So these are things based on our expertise for years with PLAN. We've also pulled them from other what are called common data elements in the literature. And then we got input from other experts, and we also piloted them with a small number of PLAN families. But these milestones are things that we think are key in the development and progression of PLAN. So it'd be something like getting a G-tube, ordering the first wheelchair, having an eye exam that showed optic atrophy for the first time. I think you all can imagine, and those of you in the study have seen what we think some of those key things are. So we also ask parents and guardians to complete what's called a patient-reported outcome measure. In this case, it's really a parent-reported outcome. They do it by proxy. And these are selected, as I said, things that are relevant to PLAN, that are standardized, and that allows us to score them reliably. So for any one child, we could look over time at how their score changes. We can look in aggregate at people with INAD or juvenile PLAN and compare, or we can compare it to a control group of people who don't have PLAN. So this is an example, a screenshot of what it actually looks like on our website. We have a portal, we send you a link, it's secure, it's on an OHSU server using a platform called RedCap. So when you go in, you see this set of probably about 25 yes or no questions, and there are just 3 of them here, and you just click yes or no. So we've tried to make it user-friendly, we've tried to make it pretty easy, and I can imagine that for somebody who does this every 3 months, it probably does get a little old. It's important to us. And what you can imagine is, sometimes for several, what we call virtual visits, we'll see sort of the same answers coming through, but then something will convert. There'll be a new milestone, something will change. And what we really want to capture is exactly when that happened and how old your child was, so we can start comparing and collecting data for the group. This is an example of what one of the parent-reported outcome measures looks like on our platform. And this is the Infant and Toddler Quality of Life Questionnaire. So it's for the little kids with INAD. And you can see, along the left side, there are things that are relevant to this population, things like how they're sleeping, how they're feeding, whether they're rolling over, sitting up, playing. And the question is, how much are they limited by their disease? Is it a lot? Is it not very much? Or is the question not even applicable at this point in time? And again, I think it's probably a pretty quick thing for parents and guardians to scroll through and complete for us. So we do also ask for healthcare providers to get involved. This can be a neurologist, a geneticist, a developmental pediatrician, but most often it's a physical or an occupational therapist, because they're the ones seeing the kids regularly and sort of familiar with their skills and their challenges with motor skills and fine motor, that sort of thing. So a lot of the questions that we ask them do overlap with what we ask parents, but they're also expanded. So things that are a little more granular and a little more technical, like discerning between dystonia and spasticity and rigidity, or hypotonia, things that we wouldn't feel comfortable asking parents about, even though most of you could answer those just fine. We want to get that from the physical therapists. And we do ask families to help drive this process by introducing me, and signing off on a release so that I can work with them directly to get some of that information on a set schedule. Statistical analysis. So we're in our 4th year of this study. We have a lot of data. I'm going to give you an update in a minute on how many people are enrolled and where we're at. But I did want to spend a minute on statistical analysis because it's important for the success of using the data and publishing it and being able to leverage it for that foundation for a trial that I mentioned. So we're using sort of a progressive model. It's called structural equation modeling. I think I've mentioned it to this group before. It's a way to model disease progression. And what it lets us do is examine different rates and patterns of disease progression at both a group level and an individual level. So we can take all those milestones and all those developmental and disease changes that I've told you about, try to determine which ones might be most important, and also look at which ones we have the most consistent and largest data set for. And then the statisticians literally build a model for PLAN, and it at an individual level, I think of it like a growth curve. When you go to your pediatrician's office, you see that paper, or maybe it's on the computer screen now, with the blue curve and for height and weight. Usually a child stays on their curve. If they're 25th percentile, they usually chug right along and stay on that line. So this is like building a model like that for PLAN. And similarly, a child would progress through PLAN sort of on their line. However, if we had an intervention, when the gene therapy trial moves into humans, that's a potential time when they could hop off that line if the intervention is working. So that's sort of a simplistic way to think about the statistical modeling that we have a contracted group doing for us, because we don't do that ourselves. And we've also learned from an interim analysis that in order to have the best power for this type of study and modeling, we ideally would have about 40 or more subjects enrolled with INAD. And, ideally that would be the same for juvenile, because we'd like to keep those two groups separate. That's a really tall order for the atypical juvenile cases, because they're even more rare. So we do have backup plans in that case where, if we don't get 40, okay, well, here's what we do if we only have 30. There's a slightly different way to do this type of modeling. But for the best power, we'd like to have 40 plus. So here's our current status. Right now, overall, we have 41 subjects enrolled. 29 of those have the infantile form, so 29 have INAD. So to get to that, goal of 40, we need about 11 more in the next year, which is a bit of a tall order. We have 12 juvenile cases. And then it's a coincidence that down below here, the 29 and the 12 happened again. I just want to tell you about our breakdown. So we have, of the 41, 29 total from the US, the UK, Australia, or Canada. I point those out because their medical records are in English and their physical therapists speak and write and use English in their records, which makes it a lot easier just from the standpoint of logistics of participation. So those people are all fully engaged, and their therapists are generally engaged as well. Some of you, I should mention, are no longer doing physical or occupational therapy for a variety of reasons, and that's okay. In that case, we just let that part drop off from what we collect. And then we do have 12 participants from other countries that are not primarily English speaking, where their medical records might be in a different language, their therapists speak other languages. In that case, if the parents themselves are fluent, that's okay. That's all we really need to get the basics done, and they keep doing those 3 to 6 months visits for us. The next 3 slides are very pretty, and I have Katrina and Puneet to thank for that. They just pulled a little more of the demographics for us to look at. So ethnic breakdown, maybe not surprisingly, this is a predominantly Caucasian group, but we do have other ethnicities represented, and 7% of our kids and adults are Hispanic or Latino. We have 11 different countries represented. And we have people from 16 different states, including our wonderful state of Oregon, which is actually pretty rare for NBIA. So just a few more things I wanted to touch on that are related to this study. One is in 2020, so last year, I think we started pre-COVID, but wrapped this up pretty early in the pandemic. We contributed a pretty big data set to Dr. Manjukurian's group, who we collaborate with frequently and openly back and forth. The data is de-identified, so any of you who are in the study, that means we scrubbed any identifier like a name or date of birth off, and we just gave it to Dr. Kurian with a coded number, so she can't link it back to any names, which is important whenever we share data. But this was to support her preparation to move into humans with gene therapy trial that she's been working on in mice. And in this case, the study she designed was retrospective, meaning they have a questionnaire and it looks back in time for people with INAD. When did this happen? When did that happen? So we were able to contribute, I think, about 20 cases to that. In addition, her funding agency, as well as the equivalent to the FDA in her country, have stressed the importance of a prospective study with any kind of move into human gene therapy trials. And that's what we bring to the table as well with this PLAN Ready. It's prospective, meaning we're capturing things in real time, marching along through 5 years, which is considered to be more reliable and more consistent than trying to glean it from medical records or look back. So both of our teams are anticipating that this is going to be an essential resource, both for her trial that'll be coming up hopefully in the next several years, and future other trials and collaborations. And then finally, I just wanted to briefly mention tissue banking. This is not specific to PLAN Ready. This is sort of a more general part of our program, but there are parts relevant to you that I wanted to just mention. So we have a partnership with the Oregon Brain Bank. This is a longstanding partnership. And we've collected both frozen and fixed brain tissue from tens of people with NBIA now, but 5 with PLAN, including 2 in pretty recent years. And that's pretty significant. It's a difficult decision for families. I think it's often a very fulfilling decision. They feel strongly about it. I hope that more people will consider it and that people will talk about it openly if you've been involved in that. But these brain tissue samples are truly priceless to us and to other investigators. There are certain questions that you can only answer with that type of tissue. And it is something that we take very good care of because we know how important it is, but we are willing to share to another investigator who has a really compelling study or something that they're trying to do. And then we have cell lines as well. So you may have been involved in this yourselves at past, not virtual, but in-person NBIA International Family Meetings. We tend to collect both blood and skin samples when we can, and we can take those cells and treat them in a certain way where we can freeze them and then bring them back. Years from now, we can bring the cells, grow them back up and use them. So these are just a wonderful resource to have in the freezer. That way, if you have a new research question that comes up in a few years, if a collaborator, if another investigator needs them, we share them openly and freely. And I think there's opportunity there for us to build up more cell lines, especially the fibroblast lines, which are useful. And hopefully at the next meeting, which will be in person, we can meet with you all and perhaps collect some more of those. So that's it for the formal part of my talk. I believe I'll be available live for questions after this concludes. And I want to thank everybody at OHSU on my team, as well as INAD Cure, for supporting specifically this study for the last several years. Thank you.
Thank you. That was a wonderful talk. I'll encourage everyone again to type your questions in the chat while people are maybe thinking. I have a question because I am new to this space and it sort of came up in another session. Perhaps you could expound upon how someone might get involved in clinical trials, or if it is for them. Some people I've seen on here are new to this world. How might someone get started with this?
Yeah, I think, there's sort of a learning curve with an ultra rare disease like PLAN. And I've been watching parents go through that learning curve for 18 years. And it's pretty intense at first. What happens is you get this new diagnosis and you get to Google and you go nuts and kind of try to identify who the players are and what's going on and who to contact. And I usually hear from people at some point in that process. But I think just getting plugged in with the community is really important. So getting plugged in with INAD Cure, getting plugged in with NBIA Disorders Association. If you're ready, entering the private Facebook group for PLAN. Some people are not ready to hop in with that. It can be pretty overwhelming, and that's okay too, to take it slow with some of these steps. But by joining the community, you're going to hear about more. You're going to learn about other people in clinical trials and their experiences. And then there are more formal ways to do this as well. Clinicaltrials.gov is always a reliable place to look for new clinical trials. Susan Hayflick made a joke yesterday about how she hadn't been there today, because things can change overnight. You don't need to look at it every day, but you could check it out every few months and see if there's anything new going on in PLAN or a related disorder. You can also check our website. We and our Facebook and Twitter, we post things that we feel are important to the community. So I think by being plugged into the general community, you've got lots of safety nets where you're not going to miss something.
Great, thank you.
Yeah.
So we will move on to our next speaker, but we'll have questions at the very end for everybody. We'll have it open. So if something pops into your head, just write it into the chat. So our next speaker is Dr. Daniel Paul. He's the Senior Vice President of Discovery and Platform Development at the New York Stem Cell Foundation Research Institute. He oversees the foundation's Global Stem Cell Array, which is an automated platform for large-scale derivation, genome editing, and differentiation of human-induced pluripotent stem cells, which he will probably be telling us all about. He's also leading efforts to develop novel approaches for detecting disease signatures and then identifying therapeutic candidates in order to treat a range of diseases, including Parkinson's, MS, and rare disorders. So I will hand it over to Dr. Paul, and again, feel free to write questions in the chat box. Our speakers are very friendly.
Thank you very much for the introduction. That segue from Allison at the end there, I can't stress enough the access to patient material, as I hope you'll hear about in a second, is key to unlocking a lot of hopefully treatments and therapies for diseases such as this. And it's great to see this happening in multiple places. So let me share my screen and right here. So, oops. Sorry, one moment. Let me just do that one more time. Sorry. Yes. Okay. I hope everyone can see my screen. Okay, so my name is Daniel. As was mentioned, I'm from the New York Stem Cell Foundation. I head up a platform here that sorry, there we go. I head up a platform here that is using stem cells to understand diseases such as INAD. And for those of you not familiar with us, the New York Stem Cell Foundation is based in New York City, and we have an overarching mission of accelerating cures for the major diseases of our time through stem cell research. And we do this in a few different ways. We have innovators that we fund around the country. They're both in the stem cell field, but we also have a neuroscience track as well, and doing early to translational research. We do a lot of outreach, obviously virtually over the past year or so, but we host a big annual conference that brings the stem cell field together. And we also host symposia and outreach, and particularly education as well. We've done a lot this year to reach out to K through 6 to kind of bring them into this space. So here we go. And then obviously we have the Research Institute as well, and I'll talk more about that in a moment. We're doing a range of different diseases here we have a range of different diseases that we study here. It ranges from diabetes, cancer, neurodegeneration, but also obviously these rare diseases as well. And we've developed a lot of underlying technology here. Some of it I'll talk about today, but it's really been a space that a lot of great research has come out of, and it's become sort of not to toot our own horn, but you know, a well-respected institute that houses a large biobank that I'll touch on, and we do a lot of collaboration now with institutes around the world, both for-profit and non-profit. So a little bit of background, just to sort of bring everybody onto the same page. And forgive me if this is all well known to you. I just thought I would start sort of at the beginning. So all of the cells in our body, if you think about the multiple different types of cells that exist, we obviously have muscle, skin, bone, brain. All of that originally came from stem cells. And you know, this is the one that maybe you have heard most about, which is embryonic stem cells. And that's been a very useful model. But over the past few years, past decade or so, we've really been able to move beyond that into all sorts of new spaces. And so in our body at the moment, if you think about as we will live right now, there's adult stem cells, and they can make basically the same type of cell that's in your body, find them maybe in the muscle and places like that, and they can only really continue to repopulate that same type of cell. We have a lot of stem cells in our blood. So one well-known one is obviously cord blood, but there are populations of blood stem cells as well, and they continue to repopulate largely the blood system and keep your immune system and things like that populated. The one that's, as I said, most important in sort of our work is this pluripotent stem cell. And it's a cell that is capable of making every single cell type in the human body, whether it's an eye cell, brain cell, muscle cell, and these are unique, except the only problem is they don't exist in our body any longer. They're only present at that very early stage in an embryonic development. And so that was why embryonic stem cells were so important for such a long time. But over the past, as I said, decade or so, this has really changed and revolutionized, because a technology that was developed by a Japanese researcher named Shinya Yamanaka, for which he's already been awarded the Nobel Prize. He discovered this in about 2007, 2008, and within the span of 5 years had been awarded the Nobel Prize because everyone realized how just revolutionary this was. What he found was that if we take skin or blood cells, just like a regular tube of blood when you go to the doctor, or skin cells from a small, sort of similar to those Allison was describing, these fibroblast skin cells, and we expose them to what we call the Yamanaka factors. That's basically what we have to do, is switch on some particular genes inside the cell. It will revert them back in time from the cell that they are to this cell that we call an induced pluripotent stem cell. And this stem cell, it behaves just like, sorry, I'm just going to turn on the spotlight here. So here we go. This cell here behaves just like an embryonic stem cell. It is expandable. We can grow basically an unlimited supply, and we can convert them into any sort of cell type that we like. So just to sort of illustrate that, this is what your skin looks like if you magnify it, and here it's stained with some particular dyes, but this is what your skin cell looks like. These are called fibroblasts, and if we expose them to these factors I was just describing, we can turn them back in time into these cells here, and every little dot that you're seeing here is a stem cell, and they grow in these sorts of clusters. We call them colonies of stem cells, but it's these cells that come from this that we can turn into sort of really any cell that we like. And so at NICEF, as I mentioned, we're doing this in a number of different ways. We have a large focus on brain diseases such as Alzheimer's and dementia, Parkinson's disease, multiple sclerosis, but we're also using it in cancer and diabetes. We have a team here generating pieces of bone from this. We do a lot of work in eye disease and obviously rare diseases as well. And so, yeah, it allows us to turn them into cells such as these beautiful neurons that you see here. Obviously, we can't, unless, access to post-mortem tissue is terrific, but we can't get access to neurons or cardiomyocytes, for example, in people that are living. And so this gives us this unique ability to generate these cells. And so you see here, these cardiomyocytes beating away in the dish. And these are relatively straightforward to grow. So these pluripotent stem cells, in theory, can give rise, as I've said, to every single cell type in the body, whether it's a neuron or a liver cell, kidney cells. There are limits at the moment, and it's sort of at the state of the art as to what exactly can be done, but we do have the ability to generate really a wide plethora of cells, and in the context of INAD and PLAN, neural cells that really are known to be affected, such as dopaminergic neurons. And we refer to these cells as patient-specific stem cells. And this gets into another key component of why these cells are so important to us, is because if we think about DNA, if you untangle DNA and look at it, its base constituents, DNA is, these four letters, T, A, C, G. And what drives a disease is really, if we focus on the G and the C here, in a disease, what can happen is that the mutation occurs, and instead of it being a C and a G, it now becomes an A and a T. And if this happens, it can cause a protein to misbehave, to not have enough of it, to have too much of it, and really that can drive the disease development. It's also what gives rise, importantly, to all the characteristics that make each of us unique. So that's an important thing. There are differences amongst all of us, and some of them are obviously, things like eye color and height and things like that. But it's these key ones where obviously in a disease context that it really, really matters. And so a change in the disease can not only cause a disease, but it can also change our response to medicines as well. And so with that in mind, if we know that diseases affect people differently, and we know that drugs affect people differently, then we need stem cells from many different people in order to understand how to treat different people. There is a reason why one drug might work for me and not for you. And just as similarly in our INAD, obviously, it's been associated with many different hundreds of changes in our DNA. And so it could be that a treatment for one may work for everyone, but it could also be the case that a treatment for one might only work for people with that same change in their DNA. And so what it means is that we need many patient samples, if we can, in order to be able to understand these differences. But just as importantly, we also need people that have the healthy DNA as well. And so it could be healthy relatives, or it could be age-matched controls, and we'll come back to this in a second. And so it comes to the question of why what we're doing here is a little bit different than others that are also approaching this in similar ways. And so over the past 10 years or so, I've had the great pleasure of helping develop a platform here, and it really combines 3 different disciplines of science. One is hardware engineering. So in that we think of robotics, we think of fabrication, we think of programming, software development. We have a big computer science team. We have data scientists here. We generate a very large amount of data. And of course, we obviously have biology as well. So stem cell research at the heart of this, but the genome editing and drug screening as well that we've sort of touched on. So by bringing all of these 3 things together under one roof, we've been able to develop a platform that allows us to understand and look at the cells from many different people at the same time. And so in this short video here, you can sort of get a sense. These robots behave the same way I do when I was doing science by hand. They'll culture cells inside these plates here. The liquid that you see is basically the food for the cells. And these robotic arms move these plates around 24 hours a day, 7 days a week, whether it's taking a picture, whether it's feeding the cells, whether it's moving them from one location to another. We have this very large automated setup that allows us to do this. And so the cells live inside this object here is an incubator. It keeps the cells at 37 degrees Celsius, just like in our bodies. And the cells can be transported around, whether they're still skin cells, whether they're stem cells, or whether we've gone ahead and turned them into neurons. And so we've built these technologies to be able to do this on the robot in a way that if we have, 10, 20, 100 different people, we can have each person in a well within a dish. So each circle that you sort of see here could be a different person, and we can look at them all at the same time. The reason for wanting to use robotics was to bring a level of standardization that's otherwise quite difficult to do by hand. And so over the years, we've been able to recruit many samples and build up a biobank here. And this is a community resource that is available to researchers around the world. And it really is end to end. So we have patient recruitment. We're able to bring samples in here. We're able to collect medical records in that same sort of red cap database that was mentioned a few minutes ago. These can be skin samples, about the size of an apple seed is the amount of skin we need, or a blood draw similar to the regular blood draws that we all have. And we can track this through our system. We've built our database on top of this. We can derive not only the skin cells, but also these stem cells. We can turn them into neurons. And in a couple of hours, I'll be talking again and we'll tell you more about what we're doing with these cells, but we can repair mutations, we can screen drugs. We do a lot of quality control to make sure that everything is correct and it is what we think it is. And then we can make an inventory that can obviously be distributed for research. And so currently, as far as INAD go, we've had the great pleasure of working with INAD here, and we've been able to collect 15 different lines. In the same way that Allison was describing, everything is de-identified. We obviously don't want this to be identifiable to any singular person. So we have family groups, which is very important to us, having not only the affected child, but having parents and siblings would be fantastic as well, allows us to do this research in unprecedented ways. And so the impact is that as we collect these large numbers of samples, whether it's patient or family controls, we turn them into these skin or blood cells. They go through this process in which we convert them to stem cells. They represent then a person inside these dishes. And as I said, we can have many people in a dish at the same time. And we can look at them whether they're skin cells, stem cells, or indeed a neuron. And so importantly, for something like INAD, we can convert them to the dopaminergic neurons that are obviously play such a key role in the progression of this disease. And so I want to just touch on sort of how to be involved, as sort of Allison was touching on, the power really comes from numbers. And just as importantly, that magic number of 40 subjects. You know, likewise for us, the more we can be able to use at the same time, the better it is for developing, you know, new treatments and understanding the disease, and importantly, understanding the disease across these different changes in the DNA. And so Lena's been a fantastic go-between from the patients to us, but we also have Jeff at this end who can help facilitate this, and I can happily share these emails. And with the right consent forms in place, we can then obtain samples and pull these into our repository here. They can be made available to the wider community. They can also be kept sort of for work that we do here. It's hopefully a relatively streamlined process to be able to do this. And I see a question just came in about what if you're not located within the USA? We do indeed have the ability to collect from overseas. It's obviously a slightly more challenging route, but we have done it, and I think if you were to reach out to either Lena or the team on this end, we can sort of engage in those discussions about this. And so obviously, you know, we view stem cells here as sort of the future of medicine. It really is studying, although I say your disease and your cells, I must stress it's in this de-identified way, but hopefully what it's allowing us to do is identify new drugs. It's allowing us to understand the basic mechanisms of why the disease develops in the first place, and sort of why it progresses as it does. And then ultimately, perhaps as well, one day is cell replacement therapy. And we're already seeing these sorts of cell replacement therapies migrate to the clinic. We have actually a trial that we're setting up here in New York to look at replacing eye cells to try and prevent blindness. And there's been a team recently that have been given approval to begin trials for the replacement of cells in Parkinson's disease. So what we're talking, it sounds maybe at times futuristic, but this is happening right now, and it's really an incredible opportunity that we have to continue this. And so although I'm talking to you today, I must stress that it's a huge team of people, like I said, and it spans all these different disciplines, from scientists to engineers. And so a huge thank you to everyone on the team, and also obviously everyone at INAD here as well for enabling us to be able to do this. And yeah, very happy to take any questions that you might have.
It was wonderful, thank you. We did have a question pop up from Maureen Miller. Is there anything grandparents can contribute in terms of blood, tissue, etc.? Yeah, absolutely. Both. It's having skin cells. I'll touch on it again, sort of in an hour or so. But skin cells to us have some unique properties that allow us to look at the disease, but we can make stem cells from blood and tissue, and again, whether it's the familial relationship, that's extremely important, whether it's just expanding our diversity or expanding the numbers of folks that we have in our repository. As I said, at the moment it's sort of
We've collected from thousands of people, and that represents about 300 different conditions, but the more we can add, the better it can be, and the more we can sort of not only do research here, but again, provide to the outward community as well.
I have a bit of a difficult question, but maybe something that's on people's minds. Do you have any sense of some timeline of when the cell replacement therapy might be heading in the direction of INAD or PLAN or these other?
It is a tricky question to answer. As I said, it's rapidly progressing. There's two sort of challenges at the moment. One is the work that's been done so far to get these cells into the clinic has largely come from embryonic stem cells. And so obviously those cells are different from the person that is receiving them. It's a bit like getting a transplant. And so you have to go on immune suppression and things like that. And so we continue to work in that space. But the great hope is that you can find either a sort of universal donor. There are people out there that transplanting into one another may be possible. So there's a lot of interest in that space. But also this very personalized approach. Take a skin cell or a blood cell, convert it into the relevant cell type and back into the same person. So it is, as I mentioned, it's sort of the first promise of this is on the way. There's a team in Japan that have been transplanting cells for Parkinson's disease. And here in New York, we have a team, not part of NICEF, I should add, but collaborators with us that are well on the way to doing this. I can't give more specifics about this, unfortunately, for this disease, but if they have successes, it only benefits everybody. And so we just hope that they are successful.
Can I ask you a follow-up about that, Daniel?
Yeah.
Without making you be specific. So the examples you gave, the eye or Parkinson's, those are very targeted regions in the brain.
They are, absolutely.
PLAN affects the entire CNS and peripheral nerves. How do you address that with replacement?
This is an enormous challenge. And it's the same for, dementia and Alzheimer's when we think of it in that context as well. It's where, cell replacement therapy is obviously great to think about, but it could be that drug discovery actually provides other windows for these much more complex things where needing to replace so many cells or so many different types of cells may be more challenging. At some point down the road, it might be possible, but this is where also the drug discovery aspect of it is perhaps a more promising route in the short term. I see some of the other questions. I can rattle through them if you like. So to Abby's question about contributing samples, again, I think in the chat it was the emails were placed. So the info at INAD Cure or Jeff at NICEF, G McGrain at NICEF. If you get in touch, we can sort of help facilitate it from there. We can't, to Joseph's question, we can't really provide back information. As a researcher, these are de-identified to us, and so that's not really possible at the moment. But again, the more that it contributes to the whole, the entire repository of INAD patients, the better it is to the wider population regardless. So although we can't give back in that way, know that the benefit will benefit many more than any one person, hopefully. To the question of dopamine pathways, again, if there's a session coming up at what time is it? 9:45 Pacific time, where I think some of this will be touched on. So we've been working with researchers around the country, sending them neuronal cells or precursors to dopamine neurons, and sort of helping them differentiate them to unlock and understand what may be different in those cells. And I'll be able to go into that in a lot more detail at that session. I think actually that addresses everything. And I'm happy to put my email in here as well. If you have any other questions, I can add my email there, and do feel free to reach out as needed. Happy to answer any other questions.
And we can also ask Allison some questions too. I want to make sure if anybody had anything that popped up along the way.
That's fine. I actually want to take 1 second to thank INAD Cure. I should have done that at the end of my talk, but they, through a small grant in the beginning and then more recently through a contract, have supported the natural history and the PLAN Ready study because they recognize that it's important for the community, and I just want to thank them for that.
Okay, I can only echo that. I think it takes foundations like this to help move this research forward, and it's a huge testament to everyone involved. It's got this far.
Well, we've officially hit time. I want to make sure if anybody wants to quickly type something, you can get it in. Otherwise, thank you so much to our speakers. This was wonderful, incredibly informative. I'm glad we got some questions asked. We've got some emails in the chat box. If you want to quickly copy those, you can, and some websites. So a lot of information today, and hopefully it was informative for you. So thank you all, and I guess we will sign off.